Hello everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s Bio-NEET. In this part of videos of molecular basis of inheritance, today we are going to talk about a set of proteins and enzymes required for DNA replication and their functions in DNA replication under the heading of the machinery and the enzymes for DNA replication. Among these proteins and enzymes involving in DNA replication, the main enzymes are DNA polymerases. These DNA polymerases are otherwise called DNA dependent DNA polymerases because they involve in the synthesis of a new DNA strand based on the sequence of nucleotides present on a parental template DNA by polymerizing deoxyribonucleoside monophosphates. Let us see that. So this is a DNA strand, parental DNA strand or parental DNA template showing 3' to 5' polarity and having the sequence of nucleotides such as AT, GC, TC, A, T like this. So in order to synthesize a new strand of DNA, first DNA polymerase has to bind to this parental DNA template at 3'. This DNA polymerase, it polymerizes deoxyribonucleoside monophosphate. This deoxyribonucleoside monophosphates are the nucleotides of DNA molecule. You know very well that there are 4 types of nucleotides in DNA. Those are deoxyriboguanosine monophosphate, deoxyriboadenosine monophosphate, deoxyribocytidine monophosphate and deoxyribothymidine monophosphate. So here the function of DNA polymerase is to polymerize this deoxyribonucleoside monophosphate in order to synthesize a new strand of DNA in 5' to 3' direction according to the sequence of nucleotides present on template DNA molecule. Actually, this deoxyribonucleoside monophosphate comes from their precursors. The precursors of this deoxyribonucleoside monophosphates are deoxyribonucleoside triphosphates. As there are 4 types of nucleoside monophosphate, here also there are 4 types of nucleoside monophosphate precursors. Those are deoxyriboadenosine triphosphate, deoxyriboguanosine triphosphate, deoxyribocytidine triphosphate and deoxyribothymidine triphosphates. So these are the nucleosides having 3 phosphate groups. These are the nucleosides having 1 phosphate groups. These are the monomeric units of a DNA molecule. So in order to give these monophosphate nucleosides, this triphosphate nucleosides undergoes a dephosphorylation reaction. 2 dephosphorylation reactions, thereby the release of 2 inorganic phosphates or a pyrophosphate. This deoxyribonucleoside triphosphates, they are acting as substrates in this polymerization reaction as they are giving the monomeric units of DNA molecule. Besides acting as a substrates, these deoxyribonucleotide triphosphates also providing energy source for the polymerization reaction carried out by this DNA polymerase. What is that energy source? The energy source is this high energy rich pyrophosphate which is being utilized by the DNA polymerases for the polymerization of these deoxyribonucleoside monophosphates in replication. These DNA polymerases are highly efficient in nature. Highly efficient means DNA polymerases can polymerize a large number of nucleotides within a short span of time. Say suppose for example, if you take the length of E. coli DNA, that is about 4.6 into 10 to the power of 6 base pairs, or it consists of 46 lakhs of base pairs. So in order to synthesize this much length of DNA in E. coli cell, the DNA polymerase takes only 38 minutes. Nothing but a polymerase can synthesize about approximately 2000 base pairs per 1 second. This much faster the DNA polymerases polymerases nucleotides in order to synthesize new strands of DNA molecules. And one more characteristic feature of this DNA polymerases is they shows high degree of accuracy. High degree of accuracy. Nothing but if it is adenosine monophosphate is there on template, this DNA polymerase exactly it polymerizes thymidine monophosphate here. Okay. If there is a thymidine monophosphate on the template, a DNA polymerase exactly polymerizes adenosine monophosphate here according to rule of complementarity and base pairing. They never polymerizes a mismatched nucleotides. Nothing but they never polymerize adenosine monophosphate for cytidine monophosphate and guanosine monophosphate for thymidine monophosphate. The DNA polymerases involving in DNA replication are of 3 types in prokaryotes. Those are DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Let us come to discuss one by one. First, DNA pol 1 or DNA polymerase 1. This DNA polymerase 1 is considered to be as Korenberg enzyme. Korenberg enzyme after the name of its discoverer R.D. Korenberg. This DNA polymerase 1 in prokaryotes shows 3 types of activities. One is a 5' to 3' polymerase activity. 5' to 3' polymerase activity. This 5' to 3' polymerase activity, it polymerases the nucleotides from 5' direction to 3' direction by using a DNA template. Say suppose for example, this is a DNA strand 3' to 5' direction. A DNA polymerase 1 by its 5' to 3' polymerase activity, it synthesizes a new DNA strand in 5' to 3' direction. It adds, it polymerizes nucleoside monophosphates in 5' to 3' direction according to the sequence of nucleotides present on DNA template. Hence it is called 5' to 3' polymerase activity. The function of 5' to 3' polymerase activity of DNA pol 1 is to synthesize a short segments of DNA molecule. What is this function? Short segments, synthesis of short segments of DNAs, right? That is. And another function of this 5' to 3' polymerase activity is DNA repairs. And one more function is it involves in recombinations in prokaryotic cells. And the next activity of DNA pol 1 is 3' to 5' exonuclease activity. Exonuclease activity. Nucleases are nothing but the enzymes which digest the nucleic acids. Exonuclease is nothing but the nucleases which digest the nucleic acids from terminal ends. Okay. Here this enzyme it cleaves, it removes the nucleotides in a nucleic acid from 3' direction to 5' direction. This 3' to 5' exonuclease activity is otherwise called proofreading activity. What we call this? Proofreading activity. By this activity, DNA pole 1, it identifies mismatched base pairs during the process of replication carried out by DNA pole 3. Say suppose for example, this is a DNA strand of 3' to 5' polarity, which is a template, and here the DNA polymerase 3 is synthesizing a new strand by polymerizing nucleoside monophosphate. Say suppose for example here adenine is a nucleotide on the template, but here DNA polymerase 3 unknowingly, unfortunately, it polymerized cytosine for adenine, to which what we call mismatched base pairing. So in such cases, this 3' to 5' exonuclease activity comes into action and removes this cytidine monophosphate, which is a mismatched nucleotide here. Thereby the DNA polymerase again polymerizes a correct nucleotide, that is thymidine nucleotide. So in such a way, this 3' to 5' exonuclease activity involves in a proofreading mechanism during the process of replication. Okay, it is removing the nucleotides in 3' to 5' direction, hence it is 3' to 5' exonuclease activity. The limitations of this 3' to 5' exonuclease activity are it can remove only mismatched nucleotides. It can never remove a properly paired nucleotides. It can remove only the mismatched nucleotides, and that too it can remove only that mismatched deoxyribonucleotides. It cannot remove ribonucleotides. It can remove only deoxyribonucleotides, and it can remove this mismatched deoxyribonucleotides one at a time. It removes mismatched deoxyribonucleotides one at a time. And the third and important activity of DNA pole 1 is 5' to 3' exonuclease activity. This activity is unique for DNA pole 1. Nothing but we can find this type of activity only in DNA pole 1, not in other DNA polymerases. The name itself indicates that it removes the nucleotides from 5' direction to 3' direction, hence the name 5' to 3' exonuclease activity. This 5' to 3' exonuclease activity can remove both mismatched nucleotides and properly paired nucleotides, and this exonuclease activity can remove both deoxyribonucleotides and ribonucleotides also. And this 5' to 3' exonuclease activity can remove the nucleotides more than one at a time. In order to understand the limitations of 3' to 5' exonuclease activity and the capacities of 5' to 3' exonuclease activity, we need to go for initiation of DNA replication in brief and Nick translation mechanism of DNA repair. Let us move on to those. So this is a DNA template of 3' to 5' polarity on which a DNA pole 3 has to bind for the initiation of replication. because DNA pol 3 is a master enzyme, a hero of replication, because as it involves in the synthesis of a large part of DNA. But here there is a constraint for DNA polymerase 3 for the initiation of replication. That is, DNA pol 3, it requires a free 3' hydroxyl group. If there is no free 3' hydroxyl group, DNA polymerase 3 cannot bind to this template and cannot initiate the replication process. Here there is a specific enzyme called a primase. What is that enzyme? Primase. By its activity, it synthesizes a short stretch of RNA primer in 5' to 3' direction by using the complementary sequences of template DNA. So this primase adds what? The primase adds a primer. Primer is what? A RNA molecule, a single stranded short stretch of RNA molecule which is being synthesized in 5' to 3' direction by the action of this enzyme primase. So that primase is providing what? A free hydroxyl group at a 3' end. This is what DNA polymerase 3 requires for the initiation of replication process. So whenever this free 3' hydroxyl group is provided by the action of this primase, the DNA polymerase 3 comes into action and polymerizes the nucleotides according to the sequence of nucleotides present on template DNA strand. Here the question is, the DNA, it must have the deoxyribonucleotides, but here the DNA is having what? RNA. So this RNA has to be removed out from this double stranded DNA molecule. In this context, the 5' to 3' exonuclease activity of DNA pol 1 comes into action, which removes this properly paired ribonucleotides of primer in 5' to 3' direction like this. So what is happening here? This 5' to 3' polymerase activity is removing properly paired ribonucleotides more than one at a time, Again, in the place of this removed primer, the DNA has to be synthesized. DNA pol 3 cannot synthesize this short stretch of DNA segment in the place of a removed primer. But here the 5' to 3' polymerase activity of DNA pol 1 comes into action. So this 5' to 3' polymerase activity, by its 5' to 3' polymerase activity, it synthesizes a short segment of DNA in the place of removed primer by the activity of 5' to 3' exonuclease activity. So what is happening here? The 5' to 3' exonuclease activity is removing the primer. In the place of that removed primer, the 5' to 3' polymerase activity of DNA pol 1 is synthesizing a short segment of DNA, so that entirely a DNA molecule will be synthesized here. So what you have to understand here? 5' to 3' exonuclease activity can remove properly paired ribonucleotides more than one at a time. And to understand the another capacity of 5' to 3' exonuclease activity, that is the capacity of removal of properly paired and mispaired deoxyribonucleotides one at a time, we need to go for a DNA repair mechanism, that is Nick translation. This is the mechanism of Nick translation, which is a method of DNA repair mechanism. Here if you observe, this is a double stranded DNA molecule where replication process is over, completed. and this is the template strand of DNA, and this is the newly synthesized strand of this double-stranded DNA molecule. If you observe this double-stranded DNA molecule is having a mismatch base pair, that is AC base pair. This mismatch base pairs might be formed when a wrong nucleotide gets polymerized and escaped out from the ice of 3' to 5' exonuclease activity. So, this mismatch base pair must be repaired. In such conditions, DNA undergoes a method of DNA repair, that is nick translation mechanism. Let us see the mechanism of this nick translation step by step. It requires two types of enzymes. One is endonuclease and another one is DNA polymerase 1. The DNA polymerase 1 showing its two activities. One is 5' to 3' exonuclease activity and 5' to 3' polymerase activity. Let us see what happens first. These endonucleases, in the process of DNA repair, first they forms a small nick on the daughter DNA strand or newly synthesized strand just before the mismatched base pair by a distance of few nucleotides towards a 5' and thereby it produces two strands of daughter DNA. One is a small segment of DNA and another one is a large segment of DNA. This small segment of DNA shows a 5' to 3' polarity, and the large segment of DNA also shows 5' to 3' polarity like this. And afterwards, the 5' to 3' exonuclease activity of DNA pol 1 comes into action, where this 5' to 3' exonuclease activity removes a properly paired deoxyribonucleotide from 5' direction to 3' direction on the large segment of daughter DNA strand. So that it removes first what? Guanosine monophosphate nucleotide on 5' from 5' to 3' direction, so that it widens the nick further. Okay. This is what the function of 5' to 3' exonuclease activity in nick translation. And finally, in the third step, the 5' to 3' polymerase activity of DNA pol 1 comes into action, where it polymerizes deoxyribonucleotides which were earlier removed by 5' to 3' exonuclease activity. Nothing but the 5' to 3' polymerase activity, it polymerizes that removed deoxyribonucleotide on short segment of daughter DNA in 5' to 3' direction. Due to action of this 5' to 3' polymerase activity, the nick whichever formed earlier before GC base pair get translated by a distance of one base pair. Now the nick came between GC and AT base pairs. So by this, the alternate action of 5' to 3' exonuclease activity and 5' to 3' polymerase activity, the nick gets translated. The nick get move forward on the daughter DNA strand every time by a distance of one nucleotide. Finally, they translated the nick towards the another end of the DNA molecule. On their way of translation of nick, here they removed that mismatched nucleotide here and polymerized a correct nucleotide, thereby DNA repairing. Here the 5' to 3' exonuclease activity, 5' to 3' exonuclease activity, it can remove both properly paired and mismatched nucleotides. It can remove a properly paired nucleotides, and it can also remove the mismatched nucleotides one at a time. So the final conclusion is a 5' to 3' exonuclease activity can remove both properly paired and mismatched deoxyribonucleotides and ribonucleotides, one at a time and more than one at a time. Keep remember this. And next let us move on to the another enzyme, that is DNA polymerase 2. The DNA polymerase 2 also shows two activities, that is 5' to 3' polymerase activity and 3' to 5' exonuclease activity. And the function of this DNA pol 2 is the synthesis of short segments of DNA, because the polymerization capacity of DNA pol 2 is very less when compared to other type of DNA polymerases, so that it never directly involves in the replication mechanism, but it involves in DNA repair mechanisms. DNA repair mechanisms. And lastly, about third polymerase of prokaryotes, that is DNA polymerase 3. This DNA polymerase 3, you know that this is the hero of a replication mechanism, which is the master enzyme, because it synthesizes a large part of DNA during replication mechanism. It also shows two activities, that is 5' to 3' polymerase activity and 3' to 5' exonuclease activity. The polymerization capacity of DNA pol 3 is very high, so that it synthesizes a large part of DNA during replication mechanism.